Welcome to this video on shotgun metagenomics, a comprehensive workflow that spans from initial sampling to detailed functional annotation. This technique provides a deep look into microbial communities. Shotgun metagenomic sequencing offers an untargeted and comprehensive view into the entire microbial universe within a sample. The entire process, from end to end, includes sampling and DNA extraction, sequencing, computational processing, assembly, and finally, annotation. We begin with Phase 1, sampling and extraction, which is foundational for the entire workflow. The quality of the initial sample collection is paramount. Diverse samples, such as soil or clinical tissues, require tailored buffers and strict cold chain logistics to accurately capture the microbial community state. DNA extraction prioritizes maximizing yield and purity by overcoming tough microbial cell walls. It also focuses on minimizing host DNA, especially in clinical samples, through depletion strategies to enrich microbial DNA fragments. Moving into Phase 2, sequencing technologies, where we prepare the extracted DNA for deep analysis. Library preparation involves fragmenting high molecular weight DNA and ligating adapters for sample multiplexing. This step normalizes the DNA, making it ready for massive parallel sequencing on platforms like Illumina or PacBio. Shotgun metagenomics demands much higher sequencing depth compared to 16S amplicon approaches. This is crucial for capturing rare species and confidently assembling complete functional pathways. Next up is Phase 3, read processing and quality control, focusing on bioinformatic cleanup. The bioinformatic cleanup begins with raw reads and involves crucial steps: quality filtering to trim adapters and remove low-quality data, and host depletion to eliminate host reference genome matches. Host DNA contamination can be significant, sometimes up to 95% of reads in clinical samples. Effective computational depletion is essential to maintain the accuracy of downstream microbial profiling. We now move to Phase 4, metagenomic assembly, where short reads are stitched together to form longer sequences. Assembly algorithms use de Bruijn graphs to stitch overlapping short reads into contigs. This reconstructs meaningful genomic contexts, providing a scaffold for identifying whole genes and functional units. Binning allows researchers to reconstruct near-complete microbial genomes, or MAGs, in silico. These MAGs are rigorously assessed for completeness and contamination before being used for ecological and evolutionary studies. Assembly-based profiling, which generates MAGs, allows for the discovery of novel species and offers excellent genomic contextualization. However, it requires significantly higher computational power than read-based methods. We have reached Phase 5, annotation and profiling, the final stage where we interpret the assembled sequences. Taxonomic profiling answers the question, who is there, by mapping reads to databases, quantifying relative abundance, and identifying core microbiome composition and key pathogens. Functional annotation reveals the community's metabolic potential, answering, what are they doing? This uncovers active pathways, antibiotic resistance genes, and virulence factors by aligning predicted genes to databases. Key databases include KEG for metabolic pathways, CAZY for carbohydrate-active enzymes, and CARD for tracking antibiotic resistance genes. As sequencing costs drop and bioinformatics mature, shotgun metagenomics is fueling massive data growth in public repositories and accelerating clinical translational research.